I now call to order the lecture portion of the Society's 2537th meeting in what is now the 155th year since its founding on March 13th, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends to the lecture portion of today's meeting and tonight's lecture by Earl Miller on how thought emerges from neural dynamics and how science is unraveling the nature of cognition and consciousness. First, let's thank our sponsors. And we're grateful to PSW full year series sponsors, PSW member Mike Helton and Helton Associates. Thank you, Mike. PSW member Joe Schulman and the Schulman Foundation. Thank you, Joe. And the sponsor of tonight's lecture, PSW member Tim Thomas. Thank you, Tim. Everybody can support the society. If you would like to make donations, you can do so online using the donations button, which is hard to find. It's in the pull-down menu under members on the top ribbon. If you're interested in sponsoring a lecture or a series or a full year, let me know. I'd be glad to talk to you about it. I'm pleased to announce the following new members. First, I have to make a correction. Last time I announced somebody by the name Eddie Brown, which was incorrect. The name was Eddie Barron. He's a scientist at the Planetary Science Institute, and he learned the PSW from PSW member Kirby Runyon. Tonight's new members are Anthony Brown, that's correct, by the way, an HR coordinator at the Nuclear Energy Institute who learned a PSW from PSW member A.C. Charania. Michael Kenny, a civil and aerospace engineer who learned a PSW from PSW member Ben Anderson. Gary Gingrich, adjunct professor at Georgetown University, who learned a PSW from, quote, many Cosmos Club members, close quote. Biswas Rijal, founder and CEO of Allo, who learned a PSW from PSW member Connor Nixon. Henry Wolkoff, a physician currently an advisor to the Nuclear Regulatory Commission, who learned a PSW from PSW member Clive Baumgarten. Adam Thompson, head of product edge computing at NVIDIA, who learned a PSW from my invitation to him to speak at the Henry lecture, which he did. And he didn't actually have to join, but he kindly decided to join. Laura Kahn, a semi-retired, as she put it, policy analyst who learned a PSW from someone at a meetup group. And Meisha Raman, research specialist at the Georgetown University Brain Bank, who learned a PSW from grad school professor Larry Milstein and fellow graduate student and PSW member Sonali Kumari. And finally, Earl Miller, our speaker tonight, who learned a PSW from the invitation to speak here tonight, and some of whose interests will be clear from tonight's lecture. All new members are entitled to a signed copy of Compendium Volume 17 of the Bulletin of the PSW. And if you've not received yours and you're a new member, please come and see me after the meeting. For those who have not joined, keep in mind that all members are invited to the pre-lecture receptions and dinners with the speakers for camaraderie and interesting discussion. If you're interested in membership, you can access a membership application on the PSW website by clicking the blue Join button at the upper right-hand corner of the homepage. And if you're here in the Powell Auditorium, you can grab one of the business cards on the cocktail tables at the back and use the QR code, which will take you directly to the membership application. PSW Science membership is open to everyone with a genuine interest in science and in the free exchange of scientific information, informed discussion on scientific subjects, and the promotion of scientific inquiry. Join now, if you're not a member. Recording Secretary Scott Matthews will now present the minutes of the 2536th meeting and the 95th Annual Joseph Henry Lecture given by Michael Garrett and Adam Thompson on agentic AI and the search for extraterrestrial life, the SETI-NVIDIA collaboration at the Allen Telescope Array. Scott, stage is yours.
Good evening. On May 29, 2026, members of the Society and guests joined the speaker for a reception and dinner at 5:45 p.m. in the members' dining room at the Cosmos Club. Thereafter, they joined other attendees in the Powell Auditorium for the lecture proceedings. In the Powell Auditorium in the Cosmos Club in Washington, D.C., President Larry Milstein called the lecture portion of the 2536th meeting of the Society and the 95th Joseph Henry Lecture to order at 8:08 p.m. Eastern Time. He began by welcoming attendees, thanking sponsors for their support, announcing new members, and inviting guests to join the Society. Scott Matthews then read the minutes of the previous meeting, which included the lecture by Harris Lewin, titled The Earth Biogenome Project, Reading the Library of Life. The minutes were approved as read. President Milstein then introduced the speakers for the evening, Michael Garrett of the University of Manchester and Adam Thompson of NVIDIA. Their lecture was titled Agentic AI and the Search for Extraterrestrial Life. The first speaker was Michael Garrett, whose portion of the lecture focused on the astrophysical foundations of the search for extraterrestrial intelligence, or SETI. Garrett began by saying that SETI was looking for, quote, technosignatures, anomalies in astronomical data that might be telltale signs of intelligence in the universe. He outlined how SETI has transitioned from a philosophical pursuit into a rigorous data-driven observational science, highlighting how the scale of next-generation radio telescopes creates a critical need for advanced AI. He said that the massive data sets produced by these instruments will require new technologies and new techniques in order to detect technosignatures. He said that this transformation was largely driven by the explosive growth in exoplanet discoveries over the last 2 decades, confirming that planets are ubiquitous in our galaxy and that many reside within the habitable zone of their host stars. Garrett gave specific examples of the type of technosignatures SETI is searching for now and in the future. These included broadband radio transmissions, the traditional focus of SETI, optical or laser pulses, brief, intense flashes of light used for interstellar communication or propulsion, megastructures, anomalous transits or thermal signatures that could indicate massive engineering projects like Dyson spheres, and broadband radio leakage, aggregate radio emissions resulting from a large number of devices operating over a wide range of frequencies. He introduced the acronym BRAT for Broadband Radio Technosignatures. He emphasized the need for AI in identifying BRATs, distinguishing them from the billions of confounding faint radio sources in the universe. To find these elusive signatures, Garrett detailed the astronomical community's roadmap for scaling up its observational power. He categorized the hardware evolution into 3 phases. Current upgrades, for example, the Allen Telescope Array and the Very Large Array and the Meerkat Radio Telescope. Next generation radio telescopes, the Square Kilometer Array and the Next Generation Very Large Array, and future concepts like lunar far-side observatories. Garrett ended his portion of the lecture by quoting Frank Drake, saying, quote, "At this very minute, with almost absolute certainty, radio waves sent forth by other intelligent civilizations are falling on the Earth." The second portion of the lecture was presented by Adam Thompson, who focused on the computational architecture required to solve the exabyte scale data bottleneck in modern radio astronomy. He explained how NVIDIA and the SETI Institute are utilizing edge supercomputing and agentic AI directly at the sensor source to bypass bandwidth limitations and perform real-time signal processing and autonomously isolate technosignatures from background noise. Thompson said that the digitization of analog RF signals creates data streams at rates of hundreds of gigabits or even terabits per second. He said that it is economically and physically impossible to pipe this raw data over standard networks to centralized servers for analysis. Rather than moving the data to the compute must be moved to the data. This requires deploying high-performance computing clusters directly at the observatory or, quote, on the edge. Thompson outlined the strategy of applying edge supercomputing, streaming data directly into GPUs, bypassing CPUs, processing data sets immediately upon generation, discarding noise and forwarding only the relevant anomalies, reducing the required bandwidth by orders of magnitude. He discussed the hardware associated with this type of edge computing, including DSPs, digital signal processors, GPUs, graphics processing units, and FPGAs, field programmable gate arrays. He described some of the algorithms or operations performed by such hardware, including fast Fourier transforms, polyphase filtering, real-time RF interference mitigation, and anomaly detection. Thompson highlighted some of the advantages of shifting these workloads to GPUs. These included highly flexible software-defined processing, rapid deployment of algorithms, dynamic scaling, massive bandwidths, and the integration of AI models and signal processing on the same devices. Thompson said that because SETI is searching for the unknown, traditional machine learning is insufficient because it requires labeled training data. Thompson explained that agentic AI in this application allows for unsupervised anomaly detection, dynamic noise suppression, and autonomous telescope operation. If an anomalous signal is detected, the AI can immediately command the telescope to dwell on the source and track it in real time. Thompson ended his portion of the lecture by saying that, quote, NVIDIA is a platform company. We make tools that other people can build on top of and then use for their own science. The lecture was followed by a question and answer session. A member asked about the impact of AI on the training and education of young people in this field. Garrett responded that he sees many students using AI on a regular basis and that he was, quote, a little worried about that, end quote. He said that while he thinks that AI is a fantastic tool, dramatically increasing his productivity, he wonders whether younger people will build up the experience and familiarity with data on a deep level. A member on the live stream asked if a more advanced civilization sending radio signals to Earth could compensate for or change the Doppler shift of the transmitted signal. Garrett responded that other civilizations could be thousands of years ahead of us in technology, and that as a result, the AIs looking for technosignatures must be open to detecting anomalies that we would not expect. A member asked about, quote, model collapse, the idea that AI-generated data is fed back into an AI as training data, ultimately causing the model to, quote, generate gibberish. Thompson responded that NVIDIA seeks to, quote, connect world-class models to world-class instruments, end quote. He said that he does not have the experience to discuss what happens when AI goes wrong. Garrett responded that astronomy deals with dynamic data sets. The sky is constantly changing. He said that this ensures that AI models are constantly changing and evolving and are therefore less susceptible to degeneration. A guest asked about the possibility of transferring specific algorithms or processes from the GPUs to other hardware like FPGAs. Thompson responded, quote, the most realistic and probably best systems are heterogeneous compute, end quote, meaning that some combination of GPUs with CPUs and FPGAs will likely be required. He gave the example of an agentic AI in a high-energy particle physics experiment that requires nanosecond latency, saying that GPUs are currently not capable of such speeds, but FPGAs are. After the question and answer period, President Milstein thanked the speakers and presented them with PSW rosettes, signed copies of the announcement of their talk, and signed copies of Volume 17 of the PSW Bulletin. He then announced speakers of upcoming lectures and made a number of housekeeping announcements. He adjourned the 2,536th meeting of the Society at 10:22 p.m. Eastern Time. Temperature in Washington, D.C., 22.8 Celsius. Weather, fair. Dinner attendance, 74. Lecture attendance, in person, 126. On live stream, 40, for a total of 166 viewers. Views of the video in the first 2 weeks, 330. Respectfully submitted, Scott Matthews, Recording Secretary.
Thank you, Scott. Do we have any comments or additions, corrections to the minutes? Hearing none, I will entertain a motion by a member to accept the minutes as read. Do I have a second? All members in favor? All members opposed? Okay, minutes are accepted as read unanimously and will be posted to the website in due course. And we now turn to tonight's lecture by Earl Miller. Earl is the Picower Professor of Neuroscience at the Picower Institute and in the Department of Brain and Cognitive Sciences at MIT. He is also co-founder and chief scientist of Split Sage and co-founder of Neuroblocks. Earl's research centers on the neuromechanisms of cognition using multiple electrode neurophysiology, psychophysics, pharmacological manipulations, and computational approaches. Much of his work concerns the prefrontal cortex and its role in integrating information and engendering flexible behavior. He is particularly well known for showing how neurons in the prefrontal cortex represent learned associations, task rules, categories, and other abstract information, and the role of oscillations in working memory and attention. Earl is an author on more than 200 scholarly publications, among them An Integrative Theory of Prefrontal Cortex Function, co-authored with Jonathan Cohn, which is one of the most cited papers in the neurosciences. Among other honors and awards, Earl is a member of the American Academy of Arts and Sciences and a fellow of the American Association for the Advancement of Science. He earned a BA in psychology at Kent State University and an MA and PhD in psychology and neuroscience at Princeton University. All questions will be taken after the lecture during our usually extensive Q&A session. Earl, the stage is yours.
Good evening, everyone. Let's just jump right in. I'm going to start with a fundamental question. What is thought? Well, probably many of us are familiar with the stuff of thought. Stuff of thought are neurons. These are brain cells, and they connect to each other at junctions called synapses, where the neurons come together. And they signal one another with brief electrical impulses that we call spikes. And there's an example of that right here. Here's voltage on the y-axis, time on the x-axis, and these neurons give these little brief pulses which travel down axons, down these neural fibers, and influence other neurons to then give off one of those spikes. So a bit like dots and dashes of a Morse code system. And this cartoon kind of illustrates that. Here's the concept here is that all these neurons are connected in this giant network, and you see the spikes kind of zipping down the neural fibers, influencing other neurons to give off other spikes. So, brains have a lot of this stuff. I'll be focusing on the cortex, which is the outer portion of your brain where a lot of critical high-level cognition goes on. And your cortex alone has about 20 billion neurons. Your whole brain has about 10 times that many, but your cortex alone has 20 billion neurons. And they form 10 to the 14 synaptic connections, 10 to the 14 junctions with each other. Now, I don't know what number 10 to the 14 is, but let's just call it a gazillion. Now, at first, these neurons were poorly understood and sometimes regarded as mere building blocks. We're talking about when neuroscience first began to form, like in the late 1800s, early 1900s, neurons were poorly understood and regarded as like bricks or scaffolding. And here's an early example here where the brain was viewed as not an intricate network, but a collection of larger scale anatomical organs, much like you have a kidney or a liver or the lungs. And why did we feel that way back over 100 years ago? Because of limitations in technology. We lacked the ability to study neurons one at a time. So we naturally focused on, we as a field naturally focused on the things we could observe, which is the gross anatomy of the brain. Also, you have to start somewhere. If you're going to figure out something as complex as the brain, you start out simple and fundamental, figure out what the parts are, and then you gradually, building on that knowledge, you gradually get more and more detailed and more deep. But back then, you got to start somewhere. By the 1950s, we then developed the technology to study the electrical spiking, these little electrical impulses from individual neurons. Here's an example here of a recording from a single neuron. This is a microelectrode, and all along the length of the electrode, it's electrically isolated. But only the very tip is there a surface that can actually record changes in electrical potential. And this electrode is very close to this neuron. So we're studying the spiking activity from an individual neuron. And this was state of the art of the field from about 1950s, 60s until around the turn of the century, this century. But when we first began to eavesdrop on the electrical spiking of individual neurons, this revealed something surprising, is that neurons are not scaffolding. I don't know how surprising it was, but we learned that neurons were not just scaffolding. They're like little information processing units. And here's an example of the Nobel Prize-winning work of Hubel and Wiesel. They were recording individual neurons from the back of the brain, so-called primary visual cortex, where vision is first analyzed in the cortex. And what they found is that an example here of an individual neuron that was an edge detector. This neuron gave off these spikes like a Geiger counter whenever the animal was looking at this leftward leaning line at a particular place on the computer screen. So this neuron was essentially a feature detector, detecting that little feature of the world, and other neurons would detect other lines in other places and other orientations. So essentially what they found, evidence that the primary visual cortex, the first stage of processing back in your cortex, broke up the visual scene into little tiny line segments. And of course, these neurons detected their things, and other neurons spiked other things. Some neurons might spike to the color red or blue or a rounded shape or a corner that I'll show you in a moment. So the idea was that all the neurons in the cortex break down the world into features, and each neuron has a particular feature it's sensitive to. So this led to the classic view of the model of the brain from the 20th century, and one that still is largely dominant today, but that's beginning to change. The view of cortex was that the cortex is wired like a telegraph system with digital-like logic gates at the synapses, at these connections that they form with one another. So here's that looks like. Here's our brain here. This is the back of the brain. That's the front of the brain. And here's primary visual cortex, where vision is first analyzed at the back of the brain. And the idea is that back here in the first stage of brain processing in cortex, of visual processing in cortex, there's edge detectors, like I just showed you. These neurons that detect different little line segments of different orientations in different parts of a visual scene. And what happens then is these edge detectors then converge. They combine signals into a second stage of processing where you have neurons that are now detecting longer edges. Then those neurons converge onto another stage of processing, and now you have a third stage of processing where neurons are detecting corners. And on and so forth until you get combined neurons, combining signals, until you get some neurons at some level of processing deep into the brain that are whole object detectors. They're detectors for this entire image that the person, the subject, is looking at. Okay? And since there's 20 billion neurons in your cortex and a gazillion synapses, you eventually get circuits and neurons that are specialized for peace, love, and understanding. That was the idea, that the whole brain is this combinatorial process, integrated process, where neurons combine information, and they do so in like logic gates that act as feature detectors. Okay. And this is maybe familiar to some of you because this is how AI works. AI works by connectionism. These large language models are trained on large data sets, and they adjust their connections to find associations that capture statistical patterns in the data. That's what AI is based on, this connectionist view of the brain. But this classic model was based on a sensory motor framework. First, we're going to study the brain by figuring out how it responds to obvious things in the outside world, like line segments, or the color red, or the roundness of a ball. And you could do the same thing from the other end, too. What neurons respond when the animal moves its arm in a particular direction or grabs something. So the idea was that we're going to start on the brain's inputs and outputs and work our way in. And that makes sense. You got to start somewhere, right? You're not going to start studying the brain by studying consciousness. You're going to start by studying how the brain processes inputs and how those eventually lead to outputs. But two things changed starting around the turn of this century. One is there was advances in technology that allowed the study of large populations of neurons. Now we can study neurons not one at a time like we did for the first half century of modern neuroscience. We can study neurons hundreds or thousands at a time. And this was key because it shifted the focus from what does each neuron do, what's the function of each neuron, to what is the populations of neurons doing as a whole. So that was a significant shift. And the other shift is we began to move beyond the sensory motor framework and began to ask questions about not how the brain detects features in the outside world, but how the brain does all the internal stuff that we associate with cognition, like make decisions or learn concepts. So, for example, our laboratory and others around the turn of this century began studying what's called top-down information, sensory information. The information coming to your brain is referred to as bottom-up information. It's the raw sensory input. Top-down information is the knowledge we learn about the world, how the world works. So in one experiment, for example, we trained most of our studies were non-human primates, monkeys, and we trained the monkeys to categorize a set of computer-generated images as either cats or dogs. We had 3 prototype cats. This is a house cat, a cheetah, and a mountain lion, and 3 prototype dogs, 3 different breeds of dogs. And we used a computer algorithm to morph or blend between all the prototypes. So we created a shape space. We created hundreds of stimuli, hundreds of images in the shape space where there was smooth variation between each of the prototypes. There was a smooth space here with no boundary in the outside world. But then we taught the monkeys that there was a boundary. We drew a boundary somewhere. Here it's drawn halfway between the cats and dogs. And over a few weeks of training, we taught the monkey that any shape on this side of this boundary is by definition a cat. Anything on the other side of the boundary is by definition a dog. And we could draw this line anywhere in this space and get the same result. So now we've created an animal that learned something, some new knowledge that wasn't present in the outside world. Learned how to categorize these 2 sets of images into one thing or another, a cat or a dog. And that's what top-down knowledge is. Top-down information is acquired knowledge. And we did a whole bunch of experiments like this. We taught monkeys concepts like up versus down. We taught them to recognize small numbers 1 through 5. We also taught them things like rule learning and rule switching. So one experiment, we showed the animal 2 pictures. First we showed them 1 picture, then after a short little memory delay, showed them 2 other pictures. And we had the animal switch back and forth between 2 different rules. On some trials, in some instances, we instructed the animal to choose the picture that was the same as the one you saw before, or we taught them to choose the different one. And the point of this, the animal is not only has to process this information about the image from the outside world, the animal also had to learn these 2 rules, then keep track of which rule it was following at that moment. And again, that's acquired knowledge about the world. So across all these experiments, and there were many, not just by our lab, but others began to pick up on this. And what we did is we recorded from the prefrontal cortex. The prefrontal cortex, before I showed you primary sensory cortex in the back of the brain, we went all the way to the front of the brain, to the prefrontal cortex. That's the highest part of your cortex. It's where your executive function supposedly resides. So we thought if we're going to find the neural basis of this top-down acquired knowledge, we should go to the top of the brain where the executive is. And when we did that, we found results that were very different from the classical model of connectionism. We found that prefrontal neurons, they encoded, they gave spikes, they signaled this top-down information, but they were largely insensitive to any details about the outside world. So, for example, we found neurons that told you, that gave off spikes when the stimulus was a cat or a dog, but didn't signal anything at all about which cat or which dog it was. Nothing about the individuals, just about the categories. Or we found neurons that kept track of which rule the animal was following, but didn't care which picture the animal was applying the rule to. So essentially what the prefrontal cortex did is it distilled all the top-down information, the rules the animal was using to solve the task, the acquired knowledge, and boiled away all the sensory details. And was not predicted from connectionism. From connectionism, there should have been developed this set of highly specialized feature detectors, not only detected cats versus dogs, but which cat, which dog, what the animal was thinking about or applying these rules to. And we also found that this top-down information, things like cat versus dog or small numbers or these rules the animal had learned, they engaged a large fraction of neurons in the prefrontal cortex. About 30 to 40, for any one of these studies, we found around 30 to 40% of the neurons signaled cat versus dog or the rule choose same versus choose different. That was, a third to nearly half of the population of neurons. And this was really something that was not predicted by connectionism at all. It was a bit of a shock, because in connectionism was supposed to build knowledge until you reach a group of neurons at the highest stages of processing that it becomes specialists. So it should be just a few specialists up there in the prefrontal cortex. Instead, like a third or 40% of the population, nearly half the population of neurons we studied are engaged by this newly acquired knowledge. And when I first presented this work about almost 30 years ago now, we got a lot of skepticism, because if neurons are supposed to be specialized, which is what everybody thought at the time, how can this possibly be? If every neuron is specialized, are you telling me, and this is a question I got when I gave my first talk on this, are you telling me the animal's brain fills up after you teach it 3 or 4 tasks? I mean, if you're devoting a third of the population to this task, what happens if you try to teach it 2 more tasks? Will the brain's capacity then get overrun? And the other thing that I heard was that, okay, well, fine, maybe it is 30% of neurons, but, these animals spent 3 weeks or a month learning these rules, like cat versus dog or numbers. They spent a few weeks doing this every single day. Maybe that's a very distorted way of studying the brain. Maybe basically the task is taken over the animal's brain by this intensive study. And I never bought that explanation, because the animals learn these tasks over about 3 or 4 weeks or a month, maybe a month and a half if they're not quite as sharp as other animals. So they're learning this over the course of 3 or 4 weeks. Well, I've been doing neuroscience for about 42 years, and I've been playing music since I was 6 years old. So that's the kind of experience we carry around in our head. The kind of experience we have is vastly more deep and extensive than anything animals in the laboratory are getting. So I never bought that explanation. So what is the explanation? Well, what I said at the time was that, well, the answer must be that these neurons are not specialists. This can't be true. You can't have a bunch of specialized neurons in cortex when you're engaging these many neurons. And so we did experiments. That's what we do. We had an idea, we had a hypothesis, we did experiments, and it turns out we were right. We were also wrong other times too, and that's when we learn more, when we're wrong about things. In this case, we happen to be right. We found that many neurons in the higher cortex were multifunctional. So now you actually train the animal on 2 or 3 tasks, like cats versus dogs, or rule switching, same versus different, or numbers, teach them to recognize small numbers. And you can keep track of the same neurons while the animals switch back and forth between these tasks. And it turns out the neurons were indeed multitaskers. They did one thing in one task and one thing in another task. Like utility players on a baseball team. You need them to do this at this time, they do it. You need them to do something else, they do something else. So neurons were multifunctional. Now, I'm going to show you what this looks like on the neuron level. And I'm going to show you what this particular task is kind of captures what these neurons are doing. This is an object sequence task, where this is, again, one of the examples how we move beyond the sensory motor framework to teach the animal something a little more complicated. In this task, you show the animal a picture, in this case, the butterfly. There's a short, like about half-second delay. Then you show the animal another picture, in this case, the padlock. Then there's another short delay lasting about 1 second. Then you show the animal 3 pictures. And you ask the animal, tell me the 2 pictures you just saw and tell me them in the right order. So what does the animal do? The animal chooses butterfly and padlock because that's what the animal saw, butterfly then padlock, just what we taught them to do. Now, according to classic connectionism, where all of cortex is a bunch of specialized neurons, what you should see is specialized neurons. You'll see a neuron in this case here. This is a neural activity on the y-axis, or how many spikes these neurons are giving off. This is time during this task on the x-axis. And here's an example of a neuron that's a single neuron that's specialized for detecting padlocks. This neuron will give the most spikes when the padlock appears. That's what the green line is. And maybe fewer spikes to the butterfly or the flower. And every time the animal sees that padlock, this neuron will respond with padlock because it's specialized for padlocks, detecting padlocks. And there might be other neurons somewhere in the brain that keep track of order, neurons that respond whenever something is first versus something is second. Okay, so all specialized neurons taking apart this task and coding it factor by factor independently. That was the prediction from the classic model of neuroscience, of connectionism. But when you actually study the brain in a task like this, you find something very different. You find neurons that show what we came to call mixed selectivity. In mixed selectivity, you get individual neurons that aren't specialized for just one thing or another, one feature or one task or one function. These neurons multiplex a wide variety of information. So, for example, this neuron I'm illustrating here responds, it shows the spikes to the padlock when the padlock appears first. But then, when you look at the object that appeared second, now the neuron is no longer a padlock detector, it's now a butterfly detector. So what this neuron is doing is essentially, it's showing a nonlinear multiplexing of wide combinations of factors. It's like a threshold detector for padlocks when the padlock appears first versus butterfly when the butterfly appears second. So what this neuron is doing is multiplexing information about the objects and which order they appear, which is the 2 factors that are relevant for performance of this task. So in other words, their spiking is context dependent. Think of this order of first versus second as being 2 different situations, like listening to a lecture here at the Cosmos Club versus being at a concert listening to music, or having dinner at home versus having dinner in a restaurant. These are different contexts. And what these neurons are doing is showing context-dependent behavior. Their spiking is context-dependent. This neuron gives different messages in different contexts. So this neuron is coding padlock in one context versus butterfly in another context. And this tells us something important because high-level cognition is context-dependent behavior. We don't react like thermostats. We don't respond to the same way, to the same stimulus, to the same sensory input every time we encounter that sensory input. We change our behavior depending on the situation at hand. And what these neurons are doing are showing this embodiment of cognition, context-dependent behavior, flexibility, which is what cognition is all about. And this makes them multifunctional. And again, these neurons, they were a large proportion of the population, and multifunctionality was not predicted by the classic connectionism model. And what we found, by we, I mean my colleague Stefano Fusi and Mattia Rigatti at Columbia University, working with our laboratory, found developed a computational model that illustrated how these mixed selectivity multifunctional neurons are essential for high-level cognition. What they do is they boost the brain's computational horsepower. So if you imagine of any problem being represented, information the neurons can represent is in this state space, this space where information can be projected. What these neurons do is they create a highly nonlinear, complex representational space that allows the brain to solve more complex problems. You know, if the more complex the shape is, the more detailed complex information can be represented in that space, and the more decision points you can find between different data points. Now, there's also, because these neurons are utility players, because they're multifunctional, because they're context dependent, they have access to a wide range of information, as opposed to specialized neurons, which just have access to just a little bit of information. And this allows the brain to learn things very, very quickly. And because memories, what the animals learned, is now distributed across many neurons and in a wide distributed way, this increases the brain's storage capacity. If you try to build a computational model of the brain where every neuron is specialized, you quickly run out of capacity and you limit what the model can learn. But you incorporate into your model these mixed selectivity neurons that do different things in different situations, and now you have a model that becomes essentially capacity unlimited because the information is distributed so widely. So what this means is that, according to connectionism, cortical processing is a drive towards small numbers of specialized neurons that detect things like faces or peace, love, and understanding. But what mixed activity shows is that instead information is distributed across large numbers of multifunctional neurons. So the cortex isn't like a telegraph system with a simple combination of signals until you get an answer. Instead, the cortex is like a vast collection of networks where all the networks are all lying on top of one another, sharing different nodes, and the networks there's a lot of crosstalk in the networks. Okay, so not specialized networks, but an influx of information where information is overlapping in the brain, in the cortex's networks. And Lisa Feldman and Baird and I just this year published a paper showing marshalling evidence that this is the result of something we call distributed dimensionality reduction. Basically, your whole brain is set up, your cortex, and your whole brain for that matter, is set up to reduce things down to their gist, to their essence, like this top-down information. And when your brain does this, it distributes the information over large numbers of neurons, producing these mixed selectivity multifunctional neurons. And it's all there in the brain's converging anatomy. So, and here's the challenge. Here's the real challenge of connectionism. Not that the data is different from what was predicted by connectionism, but the real challenge is, given what we now know about the brain, how could connectionism work? Because the problem is our brains, there's a mission control issue. Our brains have executive functions. We don't just respond to the outside world like a thermostat. Simple creatures can swim towards food or swim away from light. And they'll always swim towards food when food is there, and they'll always swim away from light when light is present. But our brains are much more complicated. We have executive functions. We are flexible. We behave and react differently in different situations. We can choose how to behave depending on the situation at hand, what we think is important, and what our current goals are. So we don't respond the same way to the same things. We respond differently depending on the situation at hand. And we also have an inner narrative. We don't just react to inputs. Our brains have this constant autobiographical narrative going on where we think, what am I doing here? Why am I doing it? What's important? What am I going to think about next? What am I thinking about now? This puts us in context and makes us make sense of the world. We know where we are and what's expected of us at any given moment. So our brains have this inner narrative that's constantly running. And part of that inner narrative is we set goals and plans. We decide what we want to do now, what we want to do tomorrow, what we want to do 3 weeks from now, what we want to do 10 years into the future. This is what our brains do very well, and what our brains do far better than animal brains do. We can make long-term plans. Now, AI, by contrast, just really reacts to inputs. AI can make complex predictions of what the next word's going to be or what the next data is going to be. But AI is in the end just really reacting to inputs. It lacks that inner narrative that our brains carry around. And it seems impossibly complex to build a control system based on the classic model of connectionism. Because remember, the connectionism is all about how the neurons connect up to one another. And there's a gazillion synapses in cortex. So in order to control a system like that, under connectionism, would require a rapid reconfiguration of countless neural connections very rapidly on the sub-second level. Parenthetically, I had to label this a manual television switchboard because now when I show this picture to graduate students, they have no idea what this is, and they're shocked that we used to make phone calls that way. By the way, none of them have heard of a slide rule anymore either. No one's ever heard of a slide rule. So things get lost. But the point is that in order to make a system that works by connectionism and can control itself, you would need some sort of puppet master that could somehow shift around this countless number of connections rapidly in the brain. And that is just computationally intractable. So what's the solution? The solution, we think, is brainwaves, a signal that was largely ignored in the second half of the 20th century. Once we shifted our focus onto single neurons, people focused on the spiking activity of single neurons and ignored the bigger picture of what was going on. And that bigger picture is these spikes, these brief pulses of electrical activity in the cortex, they create ripples in the electric fields around neurons. So that I can illustrate here. Imagine these the raindrops are the spikes. Well, the spikes create ripples. They create fluctuations in the electrical environment around the neurons. Okay, and this was known for many years, long before we recorded from single neurons. The very first signal we recorded from the brain were these electrical signals from the scalp, and that's what's shown here on the right. If you put EEG electrodes on your scalp, you get these squiggly lines, and you guys are probably familiar with that. The squiggly lines are there, and if they ever go flat, you're in a lot of trouble. You're either in a coma or you're dead. So we've known for a long time that these squiggly lines, which are essentially oscillating electrical field voltage fields, oscillating electric field effects, is happening all the time. And we also knew for a very long time that these oscillations, these brain waves, they're highly associated with different levels of cognition and awareness. So, for example, when we're highly engaged and we're thinking, we're problem solving, we're processing lots of things, you get high frequency, high being above 30 hertz, oscillations, the ripples in these brain waves. Lower frequencies like 12 to 30 hertz are associated with focus and concentration. And then you get down to even lower, frequency brain waves like alpha, and that's associated with things like restful meditation. Now you get down to even lower frequency brain waves. Now you're drowsy, and we get down to like delta. Either you're anesthetized or you're in deep sleep. So the frequency of these brain waves has long been known to correlate with level of consciousness and cognition. But for many years, it was considered to be epiphenomenal, like the humming of a car engine. When we shifted our focus as a field to what individual neurons do, the attitude was like, what do we need to look at these electric fields before? If we could look at what the neurons are doing, why do we need these ripples, this humming, this fume that the engine's giving off? So they were considered to be epiphenomenal or non-functional. Now I'm going to say right here, I think we should banish the word epiphenomenal from science, and particularly from neuroscience, because we're only scratching the surface of what the brain does. So when someone comes along and says, oh, there's a signal from the brain, but it must be epiphenomenal, no one has that kind of knowledge to say a signal like that is epiphenomenal. When I hear someone say epiphenomenal, what I hear is, doesn't fit my theory. And if your theory can't explain all the observations in the data, well, it's your theory that's falling short, not that the data is wrong. But Science is done by people. Maybe many of you have read Thomas Kuhn's Structure of Scientific Revolutions. A paradigm gets a hold of the field, and that paradigm is dominant for years and years, and it often stays well past its expiration date because people are invested in what they know and how they were trained. So the shift it takes a while to get rid of a paradigm, then the shift happens. And right now in neuroscience, we are kind of at the tail end of that shift where people are now realizing that these brainwaves, these signals, are actually functional and important. Our thinking on this has changed. We now know that brainwaves and neuron spiking work together. So the electric ripples I talked about, the little fluctuations around each neuron when the neurons give off a spike, they add together and form larger waves that take on a life of its own. So here I've illustrated, there's a buoy which we'll use as a stand-in for a spike. And when these waves form, when these ripples create these giant waves, then the waves are going to push around the buoy. The waves are going to influence spiking. It kind of has to work that way because that's basic electromagnetic theory. Neurons give off these spikes when the voltage difference across their cell membranes reach a certain level. And if the electrical environment around them is constantly changing, that's going to have a big influence on spiking activity, on the ability of neurons to give off these impulses. And we now know these waves can shape when and where neurons spike. So the waves aren't necessarily generating the spiking activity, but they're shaping the spiking activity. By the waves moving up and down, they determine when a neuron's going to spike, and by determining when a neuron's going to spike, and since waves are moving around the cortex, they also determine where a neuron's going to spike. So they're sculpting the spiking activity in your brain. It's kind of like overlaying powerful radio waves onto the telegraph system I talked about earlier. We had our telegraph system with all the spiking going on, neurons communicating through these fibers that connect them together. Now with these brain waves, it's like putting these waves across the wires that shape and modulate the impulses in the wires. So it's a whole new other level of control. So to illustrate this, here's a crowd doing the wave. See, there's a sports crowd here, and the wave is going on. The wave is moving from right to left across the computer screen, right? So this crowd of people are self-organizing based on these wave dynamics. Well, your cortex does that. So here's an example of a signal from the cortex. This is recorded from a sensory cortex. And what we see here is voltage on the y-axis, time on the x-axis. Up top here are spikes from a single neuron we recorded in cortex. And down below are these brainwave signals, these longer duration, larger electrical changes in electrical voltage that form brainwaves. And as you can see, what happens is that neurons fire spikes when the wave is cresting, and they tend not to fire spikes when the wave is waning. Okay, this is more excitation when the wave is peaking, and this is less energy, less excitation when the wave is going to a trough. So naturally, spikes track the brainwave peaks. So your brain also follows the spiking, like sports fan in a crowd. They follow these wave influences. But how do we know these waves are functional? How do we know they're not epiphenomenal? How do we know they're not just the humming of the car engine? Well, we know because we do experiments, right? And one line of work that we're conducting is on general anesthesia. Now, it may be a little disturbing to know because general anesthesia has been around since the Ether Dome at Massachusetts General Hospital. First demonstration of general anesthesia in public was 1867, I believe. Someone may correct me on that, but around 1860 or so. That was the first demonstration of anesthesia. Medical doctors don't know why anesthesia makes you unconscious. They just know that it does. It works. It's like a lot of things in medicine. It works. It doesn't kill you. Let's keep using it, right? So my colleague at MIT, Emory Brown, and Emory Brown's not only a MIT professor in my department, he's also a practicing anesthesiologist. We decided to start studying this in the cortex. So here's recordings from a non-human primate. And what's shown on top this is labeled LFP, or local field potentials. They're essentially the electric fields I was talking about, the brain waves. That's just up top here. And at the bottom are spiking activity from individual neurons recorded from the same electrodes where we record the electrical waves. And what the colors show are different areas and different patches of your cortex. So for you brain aficionados, the two blue colors are from the frontal cortex. This pink color is from the parietal cortex, which is about the middle of your cortex. And the green is sensory cortex, auditory cortex. So we're trying to bracket 3 different levels of the cortex, trying to bracket the entire cortex. And what do you see? This is now when the animal is fully conscious. What you see is a lot of high frequency and low amplitude ripples and a lot of spiking going on because all these networks are connecting, operating at different frequencies from 1 hertz to 30 hertz or more. They're all intertwining together, so you get lots of complex electrical activity, including lots of spiking. But then what we did is we anesthetized the animal using a very commonly used anesthetic called propofol. And what we found is that anesthesia, the tacit assumption was that anesthesia is just going to shut off your brain, shut off your cortex. Then you'll be unconscious. Well, it turns out anesthesia doesn't just shut off your cortex. What it does is it alters the brain waves in a dramatic way. So here's what happens when the animal is now unconscious. And we see that the brain waves, the electrical waves here recorded as LFPs, they're now forming these low frequency oscillations that are oscillating about 1 hertz. Right? And the spiking activity now is following these slow fluctuations in the electric field waves. Okay? So that's pretty cool. Now, it isn't that the anesthesia shuts off your cortex. Anesthesia shifts your cortex from this high frequency mode where lots of brain waves are interacting with one another to this low frequency hum. But it doesn't quite explain anesthesia because this low frequency hum looks a lot like sleep. This is what happens when you go to sleep at night. Your brain shifts to these 1 hertz oscillations. That's called slow wave sleep. But what's different about anesthesia? Well, what seems to be different about anesthesia from sleep, that anesthesia doesn't just shift your brain to this low frequency hum. Anesthesia misaligns the brain waves. So when under anesthesia, the waves become disconnected from one another because they become what's called phase shifted. They become misaligned. So you can see that see how the spikes are all lining up at different peaks of the wave? You can see how these spikes are moving out of phase with one another. Instead of all the spikes lining up across cortex, because these waves are out of sync with one another, now these spikes are firing at this moment, these spikes are firing a short time before, these spikes are aligned with those spikes, these spikes are aligned with those spikes, so the waves are drifting in and out of phase with one another. They're no longer in synchrony, they're drifting out of phase with one another. And as a result, all the spiking cortex now becomes misaligned and scrambled. And that disconnection could cause us loss of consciousness, something we're actively investigating right now. We believe that this is not just the shift to low frequencies, but this misalignment that prevents different parts of cortex from communicating with one another, breaks communication in cortex, and renders you unconscious. Now, in the past year or so, we did these initial studies using one anesthetic, propofol, because it's one of the more commonly used anesthetics in medicine. Since then, we've been branching out to other anesthetics. We wanted to see what effects other anesthetics might have on the brain. And it turns out we're seeing these same effects, these similar effects, the shift to low frequencies and the misalignment. We're seeing it with 3 different drugs with different molecular actions. So there's propofol, which is a GABA agonist. All you know, GABA is an inhibitory transmitter in your brain. So when propofol hits your brain, it tends to drive activity down, make the brain spike less. We've also seen it for ketamine, anesthetic doses of ketamine, which has a completely different action on the brain. It acts on a different receptor, different sets of neurons, different part of your brain. We've also tested dexmedetomidine, which is not, it's more of a, it's not an anesthetic per se, but it makes you into this relaxed, sleepy-like state. You can't do surgery on somebody under Dex alone, but you can make them sedated. So it's a sedation agent. But all these things that have this dramatic effect on your consciousness, and we can go into this unconscious, sedated, or sedated mode, they all have the same effect on brainwaves, all shifting your brainwaves to these lower frequencies and misaligning them. And think about that. These are 3 different drugs with 3 different molecular actions on 3 different parts of your brain. If you talk to my more reductionistic colleagues who are studying the brain connections and neurons at a time, and you ask them, well, how do all these three anesthetics all do the same thing? Make you unconscious and allow you to then recover from unconsciousness. How do they all do that? They would have to conclude, a reductionistic person would have to conclude that they do it by three different actions. Okay, but we're not finding that. We're finding that all three drugs do the same thing, and now we're branching out to even more anesthetics. And what that means is that we're seeing an explanation for the convergent effects of these different anesthetics. We're seeing an explanation for how these anesthetics all do the same thing at the level of brain waves that can't be explained by a more reductionistic approach. So we've explained something that the connectionism single neuron model cannot explain. And explaining something sounds pretty powerful evidence to me that this is a functional entity. Okay, so what I find exciting about this work is that if we have a universal signature of unconsciousness, it doesn't quite tell us what consciousness is, but it tells us what's required or what's different when the animal's unconscious. And we could use that as a bracket, as a guidepost to begin to study in a more detailed way the neural basis of consciousness. Okay, and that's one of the biggest mysteries of science. How is it that we're all conscious and having this experience? These kind of studies tell us which way to look. But on a more practical level, we can do something with this that helps every one of us. What Emery Brown and I are doing is we're developing a closed-loop system for automatic delivery of general anesthesia. By using EEG electrodes attached to a patient's head, we're looking for these neural signatures I just showed you as a way of detecting whether the person's unconscious or not. Now, why are we doing that? It's because anesthesia is not completely safe, especially when you're elderly, adults over the age 65. Anesthesia is linked to delirium and long-term cognitive decline. Okay, so when you get to be my age and older, you want to avoid general anesthesia at all costs. Only doing it if it's absolutely necessary because there are real cognitive deficits and delirium associated in elderly patients after general anesthesia. Despite this risk, and it's a major health issue, anesthetic dosing is guided by vital signs. Your anesthesiologist, the current medical practice, they're not monitoring the thing that's making you unconscious, your brain. They're monitoring your blood pressure, they're monitoring your heart rate, they're monitoring your respiration to see if you're reacting to the pain and make sure you're unconscious. As a result, Emery, who's again a practicing anesthesiologist, believes that many in anesthesia practice, they're giving you too much general anesthetic. They're overdosing you. They're giving you way more than you need because they're using such a crude method to determine whether you're unconscious or not. So we aim to use these neurophysiological signatures in the operating room to more precisely titrate anesthetic dose, give you just the amount of anesthesia you need and no more. And by giving you less anesthesia, we can reduce risk to these adverse cognitive outcomes. And that's something that we're currently developing right now. So another example of what these brain waves are doing is there's a divide between lower frequency brain waves and high frequency brain waves. So right now, your brain is doing something called predictive coding. You are only experiencing a small portion of the sensory information that's flooding into your brains. Your brain can't process everything that's flooding into it. So what's your brain doing? Your brain is generating predictions. Your brain is automatically generating predictions about what it's going to see or experience in the next second or so. And what the brain is doing is that anything that your brain predicts, it generates these predictions coming from the executive frontal part of your brain. Anything your brain predicts is automatically filtered out. Why? Because if your brain predicted it, it's not really telling you anything. It's not very informative if your brain can predict it. Only things that were unpredicted are passed forward as prediction errors and fully processed. And that's how you're limiting what you're experiencing to just a small fraction of the outside world. See, what's going on now is you're listening to me, hopefully. There's things in the background, the screen, the wall behind me, right? You're not actually perceiving that most of the time you think you're perceiving it. Your brain is checking every once in a while it notices the wall. Now it's making a prediction the wall is going to be there. So the whole time I'm speaking, you're focusing on me and occasionally think you're perceiving the wall. Most of that is your brain predicting that the wall is there, not actually actively processing it at the time. And the way the brain generates prediction errors is unexpected things happen or change happens. When I change the slide, you're going to notice that right away because something changed. Or just shifting my perspective is a prediction error. If I move and something new comes into my visual field, that's an error from a prediction, and that tends to get passed forward. So how does the brain do this? Well, we're finding that these prediction signals emanating from the front of the brain to the back of the brain, carrying these predictions, are carried by mid-frequency brain waves, about 10 to 25 hertz. Whereas sensory information your brain is processing is being fed forward from your brain's sensory system to the rest of the cortex, riding on higher frequency gamma brain waves. Why? Because processing information in the outside world takes energy, lots of spiking going on, analyzing that sensory information. And gamma is high energy, high frequency. There's lots of oscillations going on. There's also lots of spiking. So you have this energetic signal flooding into your brain on high frequency brain waves. But at the same time, your brain's generating predictions that are carried by these lower frequency brain waves. And they counteract the higher frequency brain waves carrying sensory information. And simply, if you just drive the brain into a lower frequency domain, you're going to get less spiking, less information. It's an automatic filter because you're now driving the brain into a lower energy state where it's spiking and processing less information. Think of it this way. Think of the gamma sensory information as spray paint that's spraying into the brain from the outside world. And what these mid-frequency alpha-beta signals are like stencils. Your brain's making predictions, making these pattern predictions using these mid-frequency alpha-beta waves. And the only thing that gets through to the rest of your cortex are whatever's missed by the stencil. Spray paint spraying through a stencil. So in other words, here's your neurotypical brain, our brains. Hopefully, our brains are generating predictions which filter out the bulk of sensory information. Only things that are unexpected or new get passed forward. And we believe this is happening, that these predictions are carried by these alpha-beta waves here. Here, just labeled as beta to save space. And they're filtering out most of those gamma waves carrying sensory information. Now, under things like ADHD or autism, the idea is that a hypothesis is that it's a prediction failure. Your brain's prediction signals are either weak or dysfunctional, and as a result, too much sensory information floods into your brain, and you get sensory overload. Well, we're finding evidence that indeed, when these prediction signals are weak, when these alpha-beta brainwave prediction signals are weak, there's too much gamma, too much sensory information flooding into your brain, and you get the sensory overload. So we believe we can record these things from the scalp using EEG electrodes or even a cap. You can wear a cap these days or a set of headphones that will detect these signals from your brain. And we believe we can develop training regimens that will allow users to get biofeedback from these signals coming from their brain and play computer games which strengthen focus. And by providing that feedback, we can strengthen the alpha-beta waves, strengthen the predictions, and mitigate the sensory overload. Now, earlier I told you about these mixed selectivity cells. That really was when a nail in the coffin, if you will, of the connectionism model. That these neurons show these multifunctional, nonlinear context-dependent spiking. Well, brainwaves can explain that too. So think of these alpha-beta stencils as being like stencils for either the concept of first versus second. So there's a different stencil. Here's our task where we're showing the animal two objects in a row. And the animals keep track of the object in their order. And we get these neurons that will respond differently to different objects, detect the padlock when it's first, but the butterfly when it's second. That's what these mixed selectivity neurons are doing. Well, we believe that's done by these alpha-beta brain waves producing different stencils. There's one stencil for first and another stencil for second. And as a result, only these neurons are allowed to respond to the butterfly. These are not because they're being blocked by the stencil. And these neurons in this patch of cortex, they're the ones that can only respond to the second stimulus, in this case, the padlock. And as a result, you get context-dependent neural spiking. You get neurons whose spiking depends on both the object and the context in which they're experienced, in this case, whether they were first or second. So it explains this mysterious property of mixed selectivity. Now, another example is showed here on this slide that these waves they're not like standing waves like a jump rope. The waves actually move around your cortex. They travel around your cortex, they swirl, they transverse your cortex, traveling waves. That's what these waves do. Now, what I'm showing here is not the waves themselves. I'm showing neural spiking here. And what I'm showing is something called subspace coding. Now, what subspace coding is, represents the activity of 200 simultaneously recorded neurons from the frontal cortex, 200 neurons. Now, if I want to describe what these 200 neurons are doing, how they're spiking, I can create a 200-dimensional space, and then each neuron would be on its own vector in that 200-dimensional space. And I can describe what those 200 neurons are doing by this 200-dimensional space. That's obvious, it's self-evident, it's self-descriptive. But then we ask ourselves, how many dimensions do we actually need to describe how these neurons are spiking, how these 200 neurons are spiking? And it turns out whether it's 100, 200, or 1,000 neurons, you can explain their spiking patterns in just 3 dimensions. So what that means is that cortical spiking is highly coordinated, not like insects buzzing chaotically in the night sky. It's more like a murmuration of birds. All these neurons are spiking in your cortex in a very highly coordinated way. Now let me go back and explain what's going on in this figure. So I'm showing here again, this is spiking patterns of 200 neurons reduced down to 3 dimensions with all the dimensions I need. So what these dimensions are essentially explain variance, how coordinated the neurons are. And in this task, what we've done is we're having the animal hold one of 4 pictures in working memory. Just like I showed you earlier, we showed the animal a picture, remember it for a second or two, then we're going to ask you about it. And what the 4 colored lines are the 4 different objects, 4 different pictures the animal's holding in working memory. And what each point in this space is, each point, ignore the motion for a moment, but each point in space is essentially a representation of what pattern of spiking is going on across the 200 neurons. So the fact that we see the separation between the 4 lines, they form in different parts of the space, means the brain is carrying information about those 4 different pictures. So what's going on with the motion? What's going on with the motion, and then in the middle of this delay, memory delay, where we're asking the animal to hold on to one of these 4 pictures, we're distracting the animal. We're showing the animal another picture, trying to knock it off its game. And it works. When you do that, when you try to distract the animal's performance drops, it reacts slower, it makes mistakes. So what's going on at time 0 is in this video, we're showing the animal the distraction. And what's going on is knocking the neurons out of neuron spiking patterns out of where they should be in this space. Then something in the brain is then not just pushing the neurons back to where they were, it's gradually steering the neurons back around to where they were before. And this is this murmuration I'm talking about. When you look at these spiking patterns in this representational space, these spiking patterns don't just shift from one part of space to another, they always move slowly throughout the space. In this case, they're rotating from where they were when the distraction appeared, and they're rotating back around to back where they need to be. Again, not a jump, but a slow rotation, like a murmuration of birds. How does the brain produce this kind of coordination? Well, we're finding that when we find these rotating movements of spiking patterns of neurons, when we record the electric fields from the brain at the same time, we find rotating traveling waves, rotating electric field waves, rotating across the surface of cortex. And what that suggests to me is that these waves are being used, much like how waves could control a crowd of people in a stadium to all act together. What these rotating electrical waves are doing are steering or herding the neurons through these different patterns. So the neurons aren't self-organizing based on shifting connections that allows us kind of high coordination. Rather, it's the electric waves themselves that produce this smooth change in neurons, in this low dimensionality space. So what this suggests to me is there's a direct correspondence between traveling brainwaves, these electric field waves that for many years people thought weren't doing anything very functional. We're now learning they are. But we're learning there's a direct correspondence between these waves traveling across the surface of cortex and spiking patterns of neurons. The things that everybody for a long time we know are very important. What neuron spiking. And what that suggests for me is there's a direct role for these brainwaves in neural computation. So it's not the humming of the car engine or the fumes given off by the engine running. They're playing an active role in sculpting what's going on in your brain. Now again, here's our crowd doing the wave here, moving from left to right across the screen. The wave's moving this way. Just by having this wave function, this wave influence, you can get a bunch of people who aren't really communicating with one another to all do the same thing in a coordinated way. And the idea is that's what your brain is doing, these electrical waves. By putting this wave of electrical activity across your brain, it's allowing the neurons to then form like a crowd of organized crowd. Okay, and these spikes are electrical activity in your brain, and these waves are also made of the same stuff, electrical activity. So when you move a wave of electrical influence across a group of neurons that are giving off electrical impulses, you're going to naturally produce an organization that reflects that wave. So, and why would the brain use this to create this kind of organization? For the same reason you can get a wave from a crowd of people who aren't talking to one another. Waves are a simple and efficient way to produce organization in a crowd. So rather than controlling all the individuals one by one, use a wave influence to guide the entire group to act in a coordinated way. So waves are not only a way to produce kind of simple organization, like I've shown you here. I showed you a rotating wave a moment ago, and here's our crowd doing the wave across the stadium here. You can also get very complex organization, not simple movements, but complex organization from brain waves. And the way to think about that is, analogy is this slide here, which shows grains of sand on a speaker. And when you play musical notes, the grains of sand form geometric shapes. Okay, so sound waves, electrical waves, are the same stuff. They're the same physics. Okay? So think of the grains of sand here as not like we're shifting around neurons in cortex. Think of the grains of sand are the spiking patterns neurons are giving off. Now we have these wave patterns that can produce complex patterns of spiking just with simple physics. So thoughts are organized. Our thoughts are organized. I'm doing my best up here. Our thoughts are organized. So spiking in your cortex must be organized. And again, it can't be by this connections in system where we're shifting connections around. Imagine the difficulty of organizing the sand grain by grain. But organization is a natural outcome of the influence of wave patterns. You get natural patterning when you have waves come together and create interference patterns. So this is basic physics that evolution could exploit, and that's what evolution does. It exploits whatever's available. And there's huge bonus features. One of them is, spiking activity, spiking communication in your cortex is actually kind of slow. If I show you a picture, it takes about 100 milliseconds, 100 to 150 milliseconds for your frontal cortex neurons to begin spiking to that picture I just showed you. 150 milliseconds is an eternity when it comes to brain processing. That's extremely slow. Spiking, these electrical wave influences, however, they travel 5,000 times faster than spiking. So the time it takes from spiking activity to go from the back of the brain to the front of the brain, electrical wave influences go back and forth 5,000 times. Okay? And if you're organizing something as complex as the brain, that kind of speed seems really useful, doesn't it? Now, another thing is that traveling waves can perform not just organize the brain, but actually perform computation. So here's an example here. Here I'm doing math here with two waves, two sine waves. There is a blue wave moving left to right across the screen, and there is a yellowish, amberish wave moving the opposite direction. So just two sine waves coming together. And what the dotted lines show is what happens when you sum those two waves. When they perfectly line up, you get a wave that's twice as big. When they cancel each other out, you get a flat line, right? And here at this point here, I've shown the sum just at this one point, but in actuality, this summation is taking place everywhere across this space simultaneously, which is an important point. So, how the waves interact determine the operation. So in this case, I'm doing addition and subtraction just by having two identical sine waves come straight at one another. You get addition and subtraction. The waves are set up because I set them up as coming in opposite directions, moving towards one another. That's how the waves are set up. How they travel together is how we model addition and subtraction. And the values are carried. The values you want to add subtract are carried in different parts of the wave. So when the waves interact, the math, all the addition and subtraction is done in parallel, all across the space simultaneously. And this is just one frequency and one dimension. Your brain could do this in multiple frequencies, anywhere from 1 hertz to 100 hertz or more, and in 3 dimensions. That's a lot of computational space to do a lot of complex mathematics. Now, this idea of waves performing complex analog computation is not conjecture, it's not hypothesis. We know it to be true. So, for example, back in 1881, there was a tide predicting machine that used wheels and disks to predict the tides. Okay, doing complex mathematics using wheels and disks. Vannevar Bush from MIT in 1927 built an analog differential analyzer that solved 18 variable differential equations using wheels and disks. Wheels and disks and oscillating electric field brainwaves are the same principles. The different parts of the wheels and disk are different variables, and how you set up the wheels and disk models the equation. It's the same principles that electric brainwaves can use. So you set the wheels, so what Vannevar would do is they would want to model an equation. They set the wheels and disks up to model that particular differential equation. And that's what took the time because you have to set the wheels and disks by hand. But the advantage of your brain could do this on the fly because it's using electric fields, not wheels and disks. So you set the wheels up to model the equation, and different parts of the wheels are carrying the different variables, much like you see here with these sine waves. And then when Vannevar and his colleagues, when they turn the crank, the equation gets solved instantaneously in one turn of the crank, or maybe 2 turns, depends on how the wheels were set up. The point is you get true parallel computing. And that makes analog computing vastly more computationally and energy efficient than digital computing. First of all, waves are information rich. When digital computers want to analyze the outside world, and again, our current model of the brain is based on the brain being a digital computer like that gives off these 1 and 0 like spikes, and then the neurons connected these logic gate like synapses. Well, when you take a digital computer takes the outside world and tries to analyze it, it's got to break apart the information into little 1s and 0s, little bits. Okay? And the outside world is continuous. The outside world is not 1s and 0s. So right there, just encoding the information is highly inefficient. The world is continuous, and analog computing works on continuous signals. But the real a big savings is from purely parallel computation. When waves interact, all the variables in the equation compute simultaneously. And digital computing is at its fundamentally sequential. You can kind of pretend that you can do parallel processing by taking 1 million processors and wiring them all together, but in the end you're all doing sequential computing. And being able to do computing in parallel is hugely efficient. So think about AI now. AI, they're now building like hydroelectric plants and nuclear power plants to power AI. Here's an example. This is a figure showing this is the energy output of the state of Virginia, I believe. I have here, state of Virginia. Right now being a few years ago, about 25% of the power that Virginia was producing was being used for AI data centers. And there's soon projected to be half the power produced by Virginia and beyond. In fact, the future energy demands of AI are likely to outstrip the ability of the world to supply it with power. Okay, so, and Microsoft just struck up a deal a few years ago to restart a nuclear power plant at 3 Mile Island in order to power a data center. Okay, so they're throwing tons of energy at AI to make it work. And AI ain't so smart. Our far more capable brains can do way more with less. Biology does not work by throwing energy at a problem. In fact, a pretty good definition of life is local reverses of entropy using as little energy as possible. Biology does not solve problems in this way. Evolution favors energy efficiency. Your far more capable brain, which is smarter than any AI out there, runs on about 20 watts of power, which is the equivalent to the power of a dim light bulb. So clearly brains are doing something fundamentally different. So what I want to conclude is that waves organize and produce thought. These signals that were ignored for a very long time are actually fundamental to how the brain does higher level computation and cognition. Brains can use AI like connectionism. Connectionism is not wrong. It's a very good model for how the brain learns and stores information. You change those synapses, you're baking information into the brain. But when you activate information and produce these electrical patterns, now the computation is done by the waves. Waves operate on the right scale. You're not trying to control the neurons one at a time, the crowd one person at a time. They operate on the right scale, the scale of crowds. Waves, by definition, operate at a level of millions of neurons simultaneously. They're a mathematically easy way to gain control over a complex system. Once you have a wave function, it's a very simple function, and once you have that wave function, you know what the action of that wave will be at a distance and over time. All with just one simple function. And they can perform highly energy efficient analog computation, allowing your brain to run on 20 watts of power. And moreover, I want to conclude that cognition and consciousness in general is analog. Analog computation allows existing wave patterns. These patterns of waves move all around your brain. And what those patterns of waves are, because they're electrical waves, they're patterns of more brain energy and less brain energy. More excitation, less excitation in your brain, which is how your brain operates. So wave patterns are the coin of the realm of how your brain is actually producing this activity. It's producing patterns of excitation and inhibition, which is what wave patterns are. And analog computing allows these wave patterns to come together and produce new wave patterns in a predictable, principled way. Now, virtually every major theory of consciousness out there, at least the ones that deal with biology, they have the same feature in common. That somehow consciousness involves your entire cortex or large parts of your cortex to become unified, get on the same page, all doing the same thing. Consciousness must be the result of unifying your cortex, organizing your cortex in a unified way. And the reason is because conscious experience is unified. Right now, you're experiencing this, what we're experiencing right now, you're experiencing it as a unified whole. The sound of my voice, what you're thinking about when I'm saying, the ambient temperature in the room, lights around you, it's all fused together into one unified experience. And consciousness, and that's not an accident. Consciousness has to be unified because while a lot of stuff goes on in your brain unconsciously, consciousness is good for mainly two things. One is planning future behavior and stopping you from doing something stupid your brain was about to do, countermanding some action your brain was about to make. And to do that, you need a big picture. You need the big picture of what's going on. And that's why consciousness needs to be a unified experience and why we experience consciousness in a unified way. So somehow consciousness is unifying the cortex. So I want to suggest that when brainwave patterns produced by analog computations become large and organized enough, when these ripples and waves come together and produce computations that then unify the cortex, much like our grains of sand here producing organized pattern across the speaker, when these patterns become large and organized, these brainwave patterns, they bind the cortex into a unified state, and the result is conscious experience. So with that, I thank you for your attention. I also especially thank our funders, and especially thank all of my students and postdocs who really do all the hard work of science.
We have time for some questions. We'll start with the red microphone.
Thanks. Matt Kaywood, member. Thank you for a very provocative talk. When you say, for example, it's been known for a long time that fast visual recognition, like a tiger in the bushes, right, is a matter of less than 100 milliseconds, and it's too fast for feedback activity. It's got to be essentially a feed-forward process. Yep. Do you believe the evidence says that that makes it an analog computation, or could it be done with some combination of analog and spikes, or spikes on the leading edge of analog wave, or how far out are you willing to get on a limb as far as it being a purely analog computation?
Well, there's going to be an analog component to all brain processing because your brain is this constant sea of rollicking electrical oscillating brain waves. So the spike's got to slot into that and slot through it and be carried forward in the brain by riding on these waves. But there's a lot of things your brain could do in a purely feed-forward way, and that's a good example right there. A bear jumps out at you, or a tiger jumps out at you, and you immediately react. That can be done just by feed-forward. It's something like a reflex, okay? But that's not cognition or consciousness. That's a reflexive reaction. In fact, it takes about half a second for your conscious mind to catch up to what's going on in the outside world. If that tiger leaped out at you and it took you half a second to decide, oh, I'm scared, I should run, you're already dead. So the way these things actually work is first you run away and then you get scared.
Sure, but you can still visually recognize it,
yeah.
And that's a computation, right? That's not just a reflex.
Yeah.
So would you say that's analog or digital or combination?
Well, I would say for both of your brain is purely digital because spikes aren't really like ones and zeros. But to the extent if we consider spikes to be a stand-in for digital computation, the waves to be something else, something beyond that kind of thing, just straight feed forward from input to output can happen in a very feed-forward way with minimal intervention from the feedback from these waves. But they're still going to be wave dependent because these spikes are being trafficked around the brain riding on these waves. But example of a quick reflexive reaction can be a purely feed-forward. I'm talking about this role in these brain waves in cognition, in setting expectations, in making predictions, in deliberation.
Blue microphone somewhere.
Yeah.
Stand up.
Oh, yes. Sorry.
Tell us your name.
Missed the first step.
Tell us your name.
Hey, I'm Christopher Schott. I'm not a member yet. I have a question related to AI and intelligence because, of course, we have to. So on the one hand, you outline how the brain reasons differently, in analog fashion, based on wave dynamics, which would make it very different from the way that AI models are currently developed. On the other hand, there seem to be fundamental mechanics here, electrical pulses that don't disclose the consciousness or intelligence emerging from a non-organic matter, say like silicon or something else. So I'm curious if you see any of your work applicable to AI development, and as a sort of consequence of that, if you could see some form of human or similar to human intelligence or consciousness emerging from machines, be they AI or others.
Intelligence and consciousness are difficult to nail down because they're hard to define. However, I will say that my point is that AI is supposed to be brain inspired, but actually only using one thing the brain does, which is connectionism, which is true. It's how the brain stores information, but it's missing all this other stuff the brain's doing, this higher level emergent property that the brain is using. But I will say this is not being missed by engineers. I'm working with engineers now who are trying to build energy efficient microchips, and doing so by not only using the digital processing of the chips like we do traditionally, but using the wave pattern, the oscillating patterns the chips will naturally generate, and use that for computation. And they're finding if you use analog computation, it reduces the power demands of the chip by 1,000-fold. So this computation, these analog computation it's having some influence on AI now because there's a real challenge with AI to make it more energy efficient, and people are now beginning to look at this kind of work as an inspiration for that.
Red microphone. Stand up. Stand up. Tell us your name.
Can you hear me? Yes, you can. You can
What's
hear me.
your name?
Dimos Kazanas from NASA. Yes.
Are you a member?
Yes, I'm a member.
Now you can ask a question.
Yes. Okay. So you gave us the waves 30 hertz, 15 hertz, and so on and so forth. Is that because have an RF circuit, it produces a wave at this frequency. And how many synapses are involved in this particular wave you showed on the
Well, the last question,
transparency?
I don't know the answer to because it's hard to calculate how many
Well,
synapses are
it's
gonna
not one, obviously.
It's not one synapse.
Yes. Is that 100 or is that a million?
Yes. No, seriously. I mean, your brain is full of what's called recurrent connections, like a snake eating its own tail. So neurons send out fibers, they snap together, and they come right back to the same neuron. That happens at a very short distance, happens at an intermediate distance, happens at a long distance. And those different lengths of recurrent cycles will naturally produce different frequency oscillations. So you get riding, so that produces these oscillatory dynamics across multiple
Feedback,
frequencies.
basically. The time to go back, basically. Is that what produces the frequency of the wave, the inverse frequency of the wave?
Well, there's two things going on. One is the recurrent connection that will naturally reverberate information, and the other thing, spiking activity, and the other thing is the EI balance, the excitatory inhibitory balance. Just by flipping the change of the balance, you can flip neurons from oscillating from low frequencies to high frequencies. So it's going to be a combination of those factors, and also in combination with things like neurotransmitters and neuromodulators, the chemistry in their brain that also affects the excitability. So there's no one simple answer to why you get particular frequencies. There's a bunch of answers.
I see. And all that propagation happens at very, very fast. You said it takes a millisecond. How much was it? 100 milliseconds,
Yeah,
basically, tenth of a
spiking
second.
to register.
But the speed in there, that happens many, many times
Yes.
during that time.
Yes. In fact, we did an experiment recently where we're recording spiking activity and electric fields from the sensory cortex. And the way people think about, even people who are who have always been sympathetic to the idea that brainwaves are doing something, they all assume that first thing that will happen is you get spiking activity. You show a picture on a screen. The assumption has been the first thing that will happen is the neurons will start spiking and they create the wave and the waves start waving.
I see.
What actually happens is the very first thing that happens, you get the brain waves. You get the high frequency gamma brain waves, and then spiking comes after that. And the reason is because the moment you look at that picture on the computer screen, neurons start spiking in your retina, and then in the next stage up. And when they create those spikes, they're already generating these electric field oscillations, and they race ahead of the spiking, almost like warming up your cortex for the spiking when it gets there. So it's a combination of the two, but because the electric field influences are faster, they actually are leading the spiking in most cases.
Okay.
Let's go to it. Let's let somebody else have a question.
Conscious and unconscious is the...
Somebody else have a question.
Sorry.
Can we go to the web, please?
We have a few questions from our online
Let's
audience.
take two from the web.
Two? Okay. Dave Rabinovitz, a member from Oregon, and Michael Haken, also a member from Silver Spring, have asked a couple related questions. First one being, how do you define consciousness? And if you characterize cognition as an inner narrative, does that imply that cognition depends on the capacity for verbal language? And have you been able to study that in nonverbal animals?
Well, let me deal with the first question first, and I want you to repeat the second one. Okay. What is consciousness? How do I define consciousness? That's a very difficult thing to define because people have been debating about this for a very long time. According to some people, the universe is conscious, and biology and brains developed evolved in order to tap into that consciousness. So that's one way of looking at it. And but people think like tables and chairs are conscious. Anything that can process information is conscious. That's what some people believe. I'm a practical person. So I think if we're going to figure out consciousness, what we should start with is defining consciousness as human consciousness. Now, obviously, humans aren't the only creatures that are conscious. But let's start there and define consciousness as human consciousness and then build out from there. Now, why do I say that? Imagine thinking like an octopus. Octopus has 8 arms, right? And people think they can do very complex kind of tasks. And people assume an octopus is conscious. And it may very well be. But the octopus nervous system is very different from ours. It doesn't have so much of a central processor like a brain we have. It has 8 separate brains on it, 8 separate arms. So if an octopus is conscious, if we somehow transmitted our minds into that octopus body, it would be a consciousness that we would not recognize as anything like ours. So I say we start with something familiar. We start with human consciousness and then build off from there. So I know it's kicking the can down the road, but that's my working definition of consciousness. And what was the second question?
Well, the second question goes along that line. If you characterize cognition, something we as humans experience, as an inner narrative, does that imply that cognition depends on the capacity for verbal language?
No, because a lot of thinking goes on that's without language. Okay, the language is extremely important and powerful because it's symbolic. If I have a word like peace, love, or understanding, that word stands in for a lot of different situations in a lot of different contexts, right? So that's a powerful way of representing information in this highly symbolic way. And that's important because your brain, your conscious mind has a limited capacity. You only really can think about one or two thoughts at a time. There's a bottleneck related to consciousness. And I think it has to do with these waves because you need these waves to come together and there'll naturally be a limiting bandwidth. Okay, but now you have, so now you have these waves going on. Sorry, what was the question again?
Oh, so,
Going off on a tangent.
if you characterize cognition as basically, like, does cognition depend on the capacity for verbal language?
Yeah, so thank you. Verbal language enhances cognition by providing the symbolic language where we could do more powerful stuff. But a lot of cognition goes on without language. I mean, it has to, otherwise people who are mute or pre-verbal children, you have to say they're not conscious. Okay, and that's obviously not true. So language is not consciousness. Language is a way of enhancing the computational power of the brain. And as such, it's related to consciousness, but it's not consciousness itself.
Okay, perfect. Thank you.
Thanks.
Okay, we're going to go to the blue microphone over there. And maybe we can get the red microphone to Federica before she shoots me.
Well, I raised my hand first.
Hello, my name is Jawad. I was a member. I'm not anymore, but I will be again very soon. So to piggyback off your the human consciousness aspect, I've been a lover and fanatic of consciousness research since I was a young, teenager. And something I realized is that Eastern philosophy, specifically around whether it's the Vedics and the Hindu side or the Buddhists, they have studied consciousness from an analytical level for thousands of years. Carl Jung talked about it in his work in his later years of his life. Now I'm realizing that a lot of neuroscience research is starting to pick up on some of the teachings or findings of mindfulness and self-awareness practices. I'm curious to know what your thoughts are about some of these deeper teachings of self-awareness through the lens of meditation, through the lens of these kind of deeper practices that humans have cultivated for thousands of years. Now the West is starting to catch up with technology.
Yeah, I don't really have much to say about that. I'm not an expert in Eastern philosophy or religions. I'm dealing with consciousness from the perspective of brain activity. So I'm, again, a very practical person. I do experiments. I look for correlations between what I see in the brain and different states of consciousness. I like, though, your definition of consciousness since you've thought about it since you were a young person. Tell us.
Well, words in itself is limiting, so you can't really define consciousness because it's purely subjective. It's like trying to describe what is love. You ask what is love to everyone in the room, we're going to have different answers. But if I were to try to define consciousness, I would define it as a field of energy, field of frequency. I think
I'm down with that.
Yeah, I think it's a field.
Let's move this to the social hour and take a few more questions from the
Thank you.
audience. The red microphone, please.
Thank you, Larry. My name is Frederica Derima, and I'm a member. Thank you for your extensive kind of work that you do in your presentation. I have many questions. One of them is, how do you train the monkeys to recognize butterfly from padlock? But maybe we can do that at the reception or the break at the end. My question is in terms of the modeling of the brain, all right? As you said, the wire model of the brain where you have neurons and synapses is not sufficient because you have the electromagnetic waves that influence other neurons and so on. And so create what you call the brain waves, what is termed the brain waves. However, the way you measure that, usually with a skull cap, which is averaging, it's not it does not, in a sense, measure what happens in between, inside the brain. Other measurements, and I'm glad you didn't mention them, is MRI, fMRI, are not sufficient also because they do measure averaging. So how we can, in a sense, create a more faithful model? I mean, slicing the brain and trying to understand the architecture of the neurons is one solution, although maybe one brain differs from the other. Maybe there are some kind of universal kind of structure in the brains. I don't know if that has been done, or and maybe it's something we should look into. So modeling the brain is an important kind of aspect. And so how we do it, given that we have the measurements that we can take are only averaging?
Well, we can make the measurements. The thing is that you're absolutely right. When you record outside the brain, look at EEG electrodes in the scalp, you're doing a lot of averaging. You're also doing a lot of filtering, because the skull and muscles filters out a lot of high frequencies. You're missing a lot. That's why we do these studies inside the brain with intracranial electrodes, so we can actually record these signals at their origin in a detailed way.
But they're only in one specific spots when you go inside the brain.
That's true. We're undersampling the brain. The brain has, the cortex has, 20 million neurons, and we only have 100 electrodes, so we do the best we can.
Yeah, are there any measurements then that we can do so we can take like CT scans of the brain that, give us three-dimensional slicing?
Yes.
Is that possible?
Technology is rapidly developing. So there's calcium imaging, so you can scan a bit of cortex very rapidly and detect activity. People are developing electrodes that record not from 1,000 neurons, but 1,000,000 neurons. So as always, technology helps us lead the way into, and when you new technology, you discover new things and make new hypotheses.
Yeah, we can discuss more at the end. Thank you so much. Yes.
Blue microphone, let me say, if you're leaving, please remember to return your name badge to the box in the back.
Ciprian Eliu Ivanov, member. When researchers use magnets to impair certain brain regions, what's happening on an electrical level in the neurons involved?
Well, what happens is you can entrain neurons by using a strong electrical pulse. Like I think you're talking about TMS, for example, where you take a magnetic pulse and you whack the brain. It's kind of like hitting the brain with a ball-peen hammer. You're changing the electrical signals right underneath where that pulse is. And that generally has either one of two things. Generally disrupts the brain because you're whacking in a way it's not used to being whacked. Occasionally you can get things like arm movement or you get a percept. That's when you hit something that's some sort of anatomical bottleneck that produces that percept or that arm movement. Generally what these pulses do is they produce a strong pulse change in the electric field, and that's generally disruptive. What I want to do is I want to not just pulse the brain like with a ball-peen hammer. I want to try to control the waves themselves, change the oscillations, so we can not just disrupt the brain, but actually try to enhance it, try to improve function. And one example I gave you is with the sensory overload. If the sensory overload is due to these prediction signals carried by this particular brainwave frequencies, particular range of frequencies, if those signals are weak, which we think they are, if we can strengthen them by gaining control of them, by reading them from the brain, then enhancing them in a selective way, then we have a chance of actually improving brain function, not just disrupting it.
We'll take one question from the web and one or two from the audience. And I think the next microphone is blue. But first, we're going to take something from the web. Then we'll go to blue.
Carl Merrill, who is a member, asks, how do you explain ambiguous figures such as the duck and the rabbit that flip back and forth as you look at the line drawings?
Yeah, there's a flipping of brainwave dynamics. And what's being said here is if I present one good example is binocular rivalry, where you present two different images to the eye, each eye, and you don't see the fused images, you see your percept flip back and forth between the two. There are studies showing that you actually see these brain dynamics flip from one percept to another. Now, the other thing we're talking about is the duck and the duck and the rabbit. right? So those are ambiguous. So that's the kind of thing that will happen. You'll see this flip in brain state related to these oscillatory brain dynamics that can flip between one percept or another. Sure.
Okay, we're going to the blue microphone.
Hi, I'm Nick. I'm not a member. So I was wondering what the spatial resolution is of the waves. Is one thing that always seemed like a problem to me about what brain waves doing much is, that seems not very information dense compared to like neuronal activity. I mean, like in the example of, you were talking about from the eye, you see something and a wave races ahead. That can tell you something's there, but how much visual information, can it give you?
That's a great point. That's what I was trying to capture with the spray paint versus the stencil. So the outside world is detailed, and that's going to be carried by these high-frequency gamma waves and spikes riding on them. So it's highly detailed information, and the higher frequency you go in the brain waves, the smaller the spatial scale of their influence. So that's why we think sensory information, the spray paint, is being carried by high-frequency brain waves because you need that fine spatial resolution to produce the kind of high-fidelity sensory analysis. We think the brain waves below 30 hertz are producing the control. They're the stencils. And that's where the brain waves are going to be particularly important, is controlling that high-frequency, fine spatial scale dynamics of the brain. Essentially, by having these lower frequency brain waves, which will then operate on a larger spatial scale than higher frequencies, that's going to be the spatial scale that you need to corral or shepherd hundreds of millions of neurons in a way that makes functional sense. And that's why we think that brain frequency range between alpha, beta, between about 15 and 30 hertz, why that's so crucial for control. We think it's because that frequency range is on the right spatial scale to produce organization in a way that will produce function.
Do you have numbers for spatial resolution?
Not off the top of my head, but yeah, they exist.
Red microphone somewhere.
Hi, Will Deacon, member. I think the work on unconsciousness is so interesting. I think like the idea that your brain kind of becomes more incoherent or slightly out of phase as you tend towards that. Have you done studies on like normal unconsciousness? Like someone who bashes their head and they go unconscious. Is that
sorry? What kind of consciousness?
If someone like becomes unconscious by non-drugs, like another means, does their brain go into the same incoherence?
We don't know that yet. What we're doing now is we're actually directly comparing sleep versus anesthesia to see if we really do see these shifts. No one's reported these misalignment of brainwaves in sleep yet. All the reported is the low frequency stuff. So we suspect when we look at actual sleep data, we won't see the misalignment, but in anesthesia-induced unconsciousness, we will. So that's ongoing work.
Super interesting. And a quick follow-up was, when you've done the injection ramps and you do like a dosage curve, does it like just onset at some point? It just like happens, or does it happen like slowly? You get these shifts.
What happened?
When you do the injection ramp, so like when you're increasing the dose every X time, does the onset of like the phase shift or the incoherence or whatever is just happen all in one go? Or
No,
it
doesn't.
Like slow and then get there?
Yeah, it doesn't. It's very interesting. You don't just see a flip from one state to another when the anesthesia hits your brain. There's this cascade of oscillatory dynamics. First, these different frequency ranges sort of bounce back and forth. Then they settle down to the state I was just talking about. And we've been focusing on just the difference between the steady state of unconsciousness versus the steady state of consciousness. We haven't been looking at the transition yet because we just started this work a few years ago. But I have a student working on those transition points right now, and especially during recovery from anesthesia, when the brain winds back up to where it was before, what's the cascade of events that leads to recovery of consciousness?
Brings it back.
Exactly.
This is the last question. I think it's the blue microphone. All right, and then the red, and then that's it.
Clive Baumgarten, a new member. A very interesting talk, thank you. Of course, the local fields that are created by the brain waves affect both excitatory and inhibitory neurons.
Yep.
How do you see that crosstalk between the excitatory and inhibitory neurons elicited by the brain waves having an effect? And my second question is, the electric fields that you're speaking of can affect spiking in two different ways. It could affect at threshold potential by altering the effective threshold potential, or alternatively, it could affect transmitter release, calcium entry, transmitter release at synapses.
Probably all of that, yeah.
Yeah, which do you think, do you have any feelings for the importance of both of those
The
different
relative importance, if I knew that, I would know a hell of a lot more about how the brain works. I think all these things are playing a factor. I'm emphasizing the influence of these electric field waves because the field's ignored them for so long. But all these things are working together. It's one system. It's spiking and waves working together. It's neurotransmitters influencing excitability, which will change the wave dynamic. It's all one integrated system.
But if you're stimulating, changing the excitability,
Yeah.
but by whatever mechanism, both inhibitory
that
and
was your first
excitatory
question.
neurons,
That was your first question, right?
how do you sort that out?
Okay, think of neurons in cortex as being like Humpty Dumpty on a wall, right? They're all teetering on the edge of spiking. Am I going to spike or am I not going to spike? Okay.
Well, they've got to be stable, otherwise they'd be randomly spiking.
What's that?
They have to have some stability around threshold potential, otherwise they'd be randomly spiking, and that wouldn't work.
No, what they're doing is they're in the state where they're just below the level of spiking. That's where most cortical neurons are resting most of the time. Then these oscillatory waves come around, so they're oscillating just below the spiking threshold most of the time. And then occasionally they hit the spiking threshold and the neuron spikes. So most neurons in cortex exist in the state where like Humpty Dumpty sitting on a wall. Am I going to spike it? I'm teetering just below it. Am I going to spike it or am I not going to spike? All change takes is a small change in the electric field environment around them to flip them towards one way or another. And by the way, one thing I should mention when we talk about spiking in the brain, how do electric waves, how they have an influence. Keep in mind one thing. Your average cortical neuron spikes at maybe 15 hertz, 15 times a second. That's well below their capacity to spike. And they give a little burst of spiking and they're quiet for a longer period of time, another burst of spiking. So just make up a number off the top of my head. Cortical neurons spike maybe 20% of the time. And neurons are only half the cells in your brain. The other half of the cells the other half of the cells are cells called astrocytes. Half the cells in your brain. They have all the electrical properties of neurons. They oscillate, they carry and respond to electrical influences, but astrocytes do not spike. So the actual situation in the electrical environment in your brain is half the cells in your brain are spiking maybe 20% of the time, but 100% of the cells in your brain are oscillating with these waves 100% of the time. So I say this to people in my field, I say, which is the tail and which is we've been treating the spiking as the dog and the tail as the waves, but it may very well be the other way around.
Last question, Tim.
Timothy Thomas, I
Red
am a member.
microphone.
Am I okay? Yeah, great lecture. Thank you very much. I'm a little confused, I'm afraid, about who's running who here. The spiking or creating the wave you're saying is creating the spiking.
Yeah.
And how are you ever going to control this wave the way we can control spiking? I mean, to get there, we have to change the electrical fields anyway, through our neurons.
Or we can induce an oscillating signal to the brain that entrains those waves and sculpt them. Now, as for the which is first spiking or waves, which is the chicken, which is the egg, that's a chicken and egg problem. So very early in development, when your nervous system is developing, it starts oscillating. That's what nervous systems do. They oscillate naturally. Any electrical system with interconnected elements that have these recurrent tail, snaking the tail-like connections, they're going to naturally begin oscillating. So very early in development, when your nervous system is first forming, it's going to start oscillating, right? At that point, it becomes a chicken and egg problem, which comes first, which is the spiking or which is the waves. They're all happening all together in mutual bidirectional influences, constantly ongoing. So the spikes create changes in the waves, and the waves create changes in the spiking.
Okay, last
a little bit in your crowd wave.
Yeah.
You could treat that as a wave, or you could treat it from a kind of a digital point where each person is responding to the next person to them.
True.
That's creating the wave.
Sure.
So
but that was
makes
just
you,
an analogy.
What?
That was just
I
an
know.
analogy.
I know. I'm using it as an analogy.
question.
Okay. So you don't really, what do you gain by not looking at it by a digital model of the crowd versus the wave model of the crowd that you seem to prefer?
what you gain is a way of understanding how the brain self-organizes itself on a level of hundreds of millions of neurons without having to do it one neuron at a time. You gain an explanation of how you have a system that can have organized self-directed cognition. So I don't see how you do it neuron by neuron and connection by connection.
We're going to save the rest for the social hour. Thank you. Let's thank our speaker here. Before you go, Earl, we have some gifts for you.
Oh, thank you.
We have a framed copy of the announcement of your talk, a rosette of PSW blazoned with the 3 stripes on the side, evocative of the symbol for inductance, which was studied by our founder, Joseph Henry, and is named for him, and a copy of volume 17, first edition of the PSW Science Bulletin. That's a compendium volume that has 3 histories of PSW, has the original, the first president's address, in which it discusses why the society was formed, what its purpose is, and it also has a discourse on how to conduct yourself at a scientific meeting, which is relevant to today. So I commend all of you to read it. And also some interesting things about talks that were given early on in the PSW's history and talks related to the same subject that were done in this century.
Thank you.
So thank you very much.
Thank you.
And there we go. We have a very few closing remarks. Our next speaker will be Doug Wallace of the University of Pennsylvania at the 2,538th meeting, 2 weeks from today on, actually 2 weeks and a day from today, on Friday, but we're back to Fridays, June 26th. That will be the last regular meeting of the fiscal year, and we'll have one meeting after that, which is technically a next year, and that will be our annual business meeting. Doug will be talking about mitochondria and their roles in health and disease. He is going to make the case that energy is important. He's been talking to NVIDIA. Yeah. I think it'll be an interesting talk. He has some reference to Chinese medicine and qi, the life force. I was a little surprised to read that, but it promises to be an interesting lecture. Mitochondria and their dysfunction is a kind of interesting and relatively recent area of study. And then finally, we have the members-only annual business meeting on July 17th, and that will end our year. So keep looking at the website to learn about what's going to come up in the fall and the spring next year. And thank you for being here. And with that, let's thank the people who run our cameras and set up and break things down. And I will entertain a motion to adjourn the meeting. A second. All members in favor? All opposed? Meeting is adjourned to the social hour.